this webinar. So I've just started recording so that we can send the file to everybody who's registered or and perhaps couldn't attend. So welcome. It's my pleasure to kick off our webinar on de novo assembly today. So we have 3 speakers lined up. I have the pleasure of going first. So I'm going to speak about single tube long fragment reads, and then Azadeh will follow me, and then Chris after Azadeh. We've tried to leave a little bit of time for questions at the end, but if you do have any questions, feel free to type them into the Q&A form I assume we have somewhere, and we'll attempt to answer them at the end as well. And if not, we'll send through a Q&A response at the end of the webinar. All right, so my name's Peter Wolf. I look after Queensland, Northern Territory and Western Australia for Decode Science. And I'd like to talk about single tube long fragment reads. So why Stlfr? So single tube long fragment reads are a method for longer reads, but using short read technology. The advantages are there's no preamplification, and we can obtain high quality whole genome sequencing libraries from small amounts of DNA input. We can achieve over 10 megabases of haplotype at contig N50 and powerful detection of structural variations such as indels, translocations, and inversions. Also, as the name would suggest, it is a one-tube reaction. It's also magnetic beads based, which means that we can automate the process. And as you're probably aware, short read sequencing is lower cost, particularly when compared to some of the longer read sequencing options available out there, and it's also low error. So what is Stlfr? So Stlfr is a code barcoding method. It was actually invented by the Chief Scientific Officer of MGI, Ray Dramatic. And as I've said already, it's a single tube long fragment read. It's basically using barcodes to add to sub-fragments of original long DNA. So we're using microbeads. There's actually millions of miniaturized barcoding reactions that occur in this single tube. We can achieve up to 3.6 billion unique barcodes, and we can apply unique barcodes to over 8 million fragments of genomic DNA from 20 to 300 kilobases in size. So I'll show you the workflow. So this is an overview, and then I'll go into more detail. So basically, we start with long fragment DNA. So we use a high molecular weight extraction kit to extract long fragment DNA. Then we do a transposon insertion. So the transposon sequence will help ligate the barcodes. So in the single tube, we'll end up adding clonal barcoded beads, our transposome, the long genomic DNA. This will hybridize the DNA with those beads with unique barcodes. Then we go through a process of ligation, PCR circularization, forming our DNA nanoballs, and then sequencing and data analysis. So I'll go through that in a little bit more detail. So for step 1, it's transposon insertion. So we start with long genomic DNA fragments, and then we combine the genomic DNA with transposome, which has 2 transposases and 2 transposon sequences. Each sequence will be inserted on the ends of the DNA. So there end up being insertions of transposon along the length of the genomic DNA. So we call this tagmented DNA. So this helps the DNA maintain a long fragment structure. In step 2, we're doing bead capture and ligation. So once the fragments have the transposon, we add the clonal barcoded beads. Each library has 3.6 billion barcodes, and each bead has about 400,000 copies of the same barcode oligo on each bead. Each sample has about 30 to 50 million barcode beads, and obviously between each barcode, these are different types of, between each bead, these are different types of barcodes. So for 1.5 nanograms of long fragment DNA, we have about 10 million molecules. So there's a higher ratio of barcoded beads to DNA. So this ensures that most barcoded beads only have one molecule of long fragment DNA. So the DNA actually ends up wrapping around the bead, being hybridized. Step 3, we do a barcode ligation. So on the surface of the bead is the barcode. We have the transpose on insertions with a capture splint oligo. This has a complementary sequence to both the barcode and the transpose on sequence. So it links the long genomic DNA to the barcode via the transposon. Then we ligate the barcoded oligo to the transpose on sequence that is bound to our long fragment DNA. So then each DNA fragment will be ligated with the same barcode for this fragment, but between fragments we have different barcodes. Step 4, we need to digest the excess oligos and remove the transposase. When we move the transposase, it actually releases the DNA, creates fragments, so we don't need to do any fragmentation per se. We then ligate an adapter for PCR, and then we do our normal PCR amplification steps. So in the case of MGI, we're doing circularization and making DNA nanoballs for sequencing on the MGI platforms. So with traditional whole genome sequencing, without the molecular barcode info, mapping can be difficult, especially with shotgun sequencing. We can try to physically separate our fragments, but it's not a very precise method. So the unique molecular barcode means we can track which long fragment the DNA came from. So the barcoding can help speed up mapping by determining where that fragment came from. So this is actual data from chromosome 11. It's a 500 kilobase DNA fragment, and it's showing the coverage that we can achieve with Stlfr. So there's 3 different barcodes shown, 56, 2345, and 144579. And we can see a number of reads that have been mapped to the same region of that chromosome. We can actually then overlap multiple locations to get even better coverage. We see about 10 to 20% of the long fragment gets the barcodes. This is better than short read technology. In terms of fragments per bead, as I said, we tend to get, in most cases, only one long fragment per bead. And we see that in about 85% of cases. So more than 85% of long genomic DNA molecules bind to a single barcode. For about 10%, we see 2 fragments, but generally on probability, these are from 2 different chromosomes. So when we do our analysis, we're able to map them back to different chromosomes. In terms of fragment length, so depending on the freshness of DNA, the average captured fragment length is between 50 to 70 kb. We do see fragments up to 300 kb, but because the beads are 2.8 micron, a 300 kb molecule needs to wrap around that bead about 10 times, so we don't generally see that many fragments at that size. In terms of just some data here on variant calling accuracy, so we can see a comparison here on the right is a PCR-free standard library and human chromosomes using Stlfr. And the sensitivity and accuracy holds up pretty well compared to that PCR-free standard library, certainly over 99%. In terms of indels, in the genie in a bottle samples performance, so against the PCR-free internal control library again, Stlfr produces accuracy and sensitivity over 96%. So in terms of de novo genome assembly, obviously longer reads are better, but single tube long fragment reads provide an alternative to 3rd generation long sequencing. Azadeh and Chris will also talk about some other options coming up shortly. So just a few more slides on data. So this is short read and ONT data for a human genome. So the config N50 and the scaffold N50 is shown. So the ONT was used to close some of the larger gaps in the assembly, and we call this the 676 standard. So we're able to achieve 10 to the 6 for config N50, 10 to the 7 for scaffold N50, and a genome size of 6 gigabases. So we can do haplotype phasing with this data as well. In terms of comparison with some of the longer read technologies, so for human de novo whole genome sequencing, STLFR and ONT, we can use a lower DNA input, so 1 nanogram, compared with 5 micrograms for the other options. In terms of cost, in relative terms, it's a much lower cost for this method compared to the long sequencing technologies, and also requires less computing cost. This is a pretty cool project that BGI helped out with. So it's a 35-day de novo project. So from sample to publication, it took 35 days to do a de novo assembly. This is on fall armyworm, which is an invasive species in China. And it was done in collaboration with the Institute of Zoology, which is part of the Chinese Academy of Science. So from day one, when they initiated the project and were starting to collect samples, they were able to publish by day 35. So by day 2, they'd already done some samples and prepared libraries for sequencing. Day 5, they had their STLFR data. So they needed to do some whole genome sequencing in RNA-seq and even some gap closing here later on in the project. But for 35 days for a whole de novo assembly is a pretty good feat. We can also work on non-human species. So this data is just shown for 30 marine species, showing STLFR assembly, and good scaffold N50s there, so in the megabase level. I know there's some plant people on the webinar today, so we're also able to accomplish de novo assembly of plants. So there have been some projects undertaken on lettuce and teak, and the config and scaffold N50s look good for those projects as well. Just a couple more slides and then I'll hand over to Azadeh. So in terms of future applications for STLFR, so MGI are quite progressive in developing their applications. They're looking at full-length mRNA sequencing with single cell technology, also deep population sequencing of 16S RNA, and phased chromatin mapping by ATAC-seq, and also methylation studies. And this is my final slide. So STLFR can provide a long fragment read total solution. From DNA input with high molecular weight DNA, there's kits available on the market for that, to library construction using the MGI Easy STLFR library prep kit, to sequencing on the MGI platforms, G50, G400. And we also provide some nice drop-in analytical tools as well, like Megabolt. And of course, if you'd like any more information on any of those topics, feel free to reach out. So I'll end my presentation here and hand over to Azadeh.
Thank you. Did you want to answer any questions that anyone might have about STLFR before we move to Dovetail?
I'm happy to leave it to the end.
Oh, okay.
Is that okay?
Sure.
Feel free to write some questions in the, I assume that we've got chat here somewhere and I can answer as Azadeh is speaking as well.
Let me see if I can share my screen. Okay. Can you please make me host, Pierre? So there's a little box with 3 dots when you hover your mouse over mine. Yes, so I guess that would be.
Okay. You should be good to go, I think, Azadeh.
Yes, yes. Hopefully you can see my screen.
Yes.
Okay, great. Okay, so good afternoon or good morning, depending on where you are. My name is Azadeh, and I work with Peter at Decode Science, looking after our customers in Victoria as well as South Australia. I'd like to thank you too for joining this webinar on de novo assembly and the different technologies that are available. And it's very common for people who are interested in de novo genome assemblies to use and combine data from multiple technologies, because it's very rare to be able to put a good quality genome together with just using one technology, especially if it's just short read sequencing technology. So it's quite common and well-established kind of procedure in the area to use multiple technologies and combine the advantages that each of them offers. So, Peter told you about Stlfr and how it helps with really the most challenging part of genome assembly, which is being able to joining contigs and scaffolds together to make the most contiguous assembly that's possible. So yes, Peter did a great job in explaining that. So I'm gonna switch gears and tell you about proximity ligation technology, and more specifically, the new assay from Dovetail Genomics, which is called OmniC. So a lot of you may already be familiar with Hi-C, with proximity ligation, and how it's applied to improving genome assemblies. So, but it's OmniC could be new, so you may not have heard of OmniC, and how it's different from Hi-C, from a traditional Hi-C, and what benefits it offers. And after me, Chris is going to talk about something, a completely different technology. And then actually at the end, we're going to have we're going to put all of these technologies together and tell you how you can actually combine these, how they complement each other, and how you can choose what's the right technology or technologies are for you. So I'm going to get started with introducing OmniC. So I'm going to tell you a little about Hi-C or proximity ligation in general. And I'm going to introduce OmniC, which is a new assay from Dovetail Genomics, and how it's been applied to improve genome scaffolding with a proximity ligation method. So as I mentioned, this is a usual process for building a reference genome. You start with a short read sequencing, Illumina or MGI, depending on budget. People have also been using PacBio, and you put this together and you come out with the contigs. And then the next step is to put the contigs together and make scaffolds. And finally, the final goal of every genome assembly is to be able to put as many scaffolds together as possible to end up with just one scaffold per chromosome. So that's the final goal of every genome assembly. So this is where OmniC comes into play in joining contigs together as well as scaffolds together. So the range of application of OmniC is both in mid-range as well as long-range assemblies. And that's something that's quite new and unique to OmniC for people who may have been already familiar or using Hi-C technologies for assemblies before. So proximity ligation technology in general works based on well, taking fresh tissues, fresh or frozen tissues of any organism that you're working with. So for plants, this could be young leaves, or for animals, it could be blood. And so in this schematic, what you see here, the dark or black lines here are DNA or our chromatin. So this is the chromatin. So black lines are DNA, and then the blue discs are histones. And this is, so this in this schematic, you can see DNA being wrapped around histones in its native form in cells. So the first step is cross-linking the cell, so that everything is stays where it is, it's fixated where it is. And you notice that there are points 1 and 2 where they actually could be hundreds of kilobases apart in a linear view of genome, but in a three-dimensional view of the genome, they actually are close together. So the next step is a digestion. So this is traditionally done by restriction enzymes, followed by end repair and ligation. So now notice that point 1 and 2 are actually in the same fragments. They are like joined together. The next step is to basically undoing these crosslinks and proceeding with making next generation sequencing libraries, followed by a short read sequencing, a paired end short read sequencing, which can read both ends. So this we can read section 1 as well as section 2, and in the same, from the same molecule. And then what this does is that when we have our draft assembly here with different scaffolds here, then we can map our paired end reads. So you can notice that one end maps to one scaffold and the other end maps to another scaffold. And now we can use this feature and join these scaffolds together. So now we can use these paired end reads from our Dovetail or from our proximity ligation assay to be able to join the scaffolds together and make a more contiguous genome. And I'm not going to spend much time in this, but just telling you that Dovetail Genomics also offers a service for data analysis called HiRise, which has a very rigorous way of combining your draft input genome with proximity ligation data to give you a HiRise scaffold assembly, a very high quality genome assembly. So if any of you is well, they haven't, they're just about to get started with proximity ligation assay based scaffold assemblies, and you don't have the bioinformatics expertise, you can look to service from Dovetail Genomics. So now I'm going to specifically tell you about OmniC as a proximity ligation assay, and then tell you how it differs from traditional HiCs. So as I mentioned before, in traditional HiCs, the chromatin digestion happens with the restriction enzyme. And that works great if you have restriction enzyme sites everywhere in the genome, which normally you don't have. So what OmniC does is to use a general DNase instead of a restriction enzyme. So now you have a lot more sites which are available to for digestion, and that's going to dramatically improve the coverage and the quality of final assembly. And I'll tell you how it happens. So this is a comparison of data between OmniC and traditional HiC with a single restriction enzyme or even multiple restriction enzyme. So the dotted lines here are Illumina shotgun data. So this is number of bases. So you can't see what's on the x-axis, sorry, but this is a coverage. So the dotted lines are the Illumina shotgun data, and the blue, red, and orange lines are data from OmniC, single restriction enzyme Hi-C, and multiple restriction enzyme Hi-C. So just you can at the beginning you can see that OmniC data mimics shotgun Illumina data a lot more closely in terms of coverage. And how it translates into giving a better genome assembly, you can see that here the even coverage across genome with OmniC. However, with Hi-C, with a single restriction enzyme, depending on whether we have any restriction sites on any area of the genome or not, we can we can see, for example, areas like here, where we have very little coverage compared with other pods because we didn't have many restriction enzyme sites here. So you can improve that to some degree by using multiple restriction enzyme sites. However, you can still see these blind spots here. So which means that some areas of genome would be inaccessible. You have to do a lot of sequencing to be able to cover all the genome. This is another data showing how scaffolding efficiency could change depending with traditional Hi-C methods, depending on how many restriction sites exist per contig. So you can see if you have, so the blue bars here are data from OmniC and the gray bars are from Hi-C. You can see that if you have at least 4 restriction enzyme sites per contig, you can have a 100% efficiency in contig scaffolding. However, if you have fewer than 3 restriction enzyme sites, then the Hi-C based methods of scaffolding start to show a dramatic decline. So if you have just one, let's say one restriction enzyme site per contig, you only have a 60% efficiency in scaffolding. And if you have 0 sites, then it's a lot lower. However, if you with OmniC, you still have a quite good acceptable efficiency of scaffolding if you don't have any sites, because it doesn't rely on any specific restriction or site for digesting the chromatin. So I'm just going to go back to this slide that I presented earlier and tell you, because I want to explain another difference between OmniC and Hi-C. So with Hi-C, because of that difference, because of that usage of restriction sites, it's mostly only usable in long range part of de novo assembly, which means that but however, with OmniC, you can OmniC helps with both mid-range as well as long-range parts of genome assembly. And what it means is that if you have a good, so you have a draft genome assembly and you have a N50 size for your scaffolds. So if your scaffolds are already long enough, which means more than 1 megabases, then you can get an acceptable improvement on your assembly if you just use Hi-C. But if it's if your draft assembly does not have longer than 1 megabase in terms of scaffold N50 size, then the Hi-C is not going to be of much help. You need a mid-range kind of technologies to put these contigs together, and improve the scaffolds N50 size before you use Hi-C. But basically with OmniC, you can do 2 assays in one go. So you can use OmniC for putting contigs together as well as scaffolds together to come up with a really good final assembly. So these are just some software that I'm going to just very briefly mention. So these are some open access softwares that you can use to analyze OmniC data. where we're talking about genome assembly today, but you can also actually use this for structural variant detection, SNP and indel genotyping, as well as haplotype phasing and looking at chromatin conformation. So, and by the way, I'm going to share the slides with you later. I'm going to email them to you so you can have these, can have a look at these, and you can ask us if you have any questions about any analysis softwares. We're happy to help you. So I'm just going to very quickly go over a case study because I'm conscious of time. So this is it looks at the genome assembly of Cheetgrass, which is not actually a native. It is not a native plant in North America. It was brought to North America from Europe, and it's a plant that grows uncontrollably in different areas, and it absorbs a lot of water, and it makes the land very dry, and it causes fires because of that. So they compared the high C versus Omni C in the final genome assembly and how and the quality of genome assembly. So this is the method that they use. They used PacBio data to put some context to generate, and then they use Dovetail High C as well as Dovetail Omni C to generate a final assembly. So both High C and Omni C result in 7 pseudochromosomes. So that's great. So at least they kind of validate each other. So you have 7 pseudochromosomes from both methods. However, when they did a self-synteny study, so that's in the x-axis you have these different chromosomes that have been put together using high C, and the y-axis are the chromosomes that have been put together with Omni C. And this graph looks at the concordance between these two chromosomes, and they just to see if they agree with each other, if they kind of validate each other. And in most cases they do. However, in chromosome 4 and 5, they detect a inversion. So something that I have to mention, and I think Chris is going to talk more about it, is that high C methods, traditional high C methods, are notorious in terms of giving false positive inversion. So basically they can connect scaffolds together, but somehow in an inverted way. So the data may not be quite accurate, but what they have done with Omni C is that they obviously detected an inversion here, and they wanted to see which one is correct. Is Omni C giving you the correct quantic orientation, or is it high C? So they looked at scan telomeric regions for these two chromosomes, and with the chromosomes put together with Omni C data, they can see that telomeric regions appear at both ends of chromosomes as expected. But in the high C data, they see these telomeric regions appear actually in the middle of chromosomes, which is not the case. Well, it's incorrect because telomeric regions are at the ends of chromosomes. So this is how you can see that Omni C can actually give a more accurate assembly by doing a better job at basically putting the context together in the correct orientation. And Chris can tell you about how you can actually apply Bionano to further help you with that. So just some information on again on bioinformatics, which I'm not going to go through. So in the interest of time, so just a little bit about, just to give you an idea of how much sequencing these libraries take. So the answer is it depends on your genome size. So this is like a guide, a rough guide for you to have in terms of how many read pairs you need depending on your genome size. So, so this is the genome size in megabases and million read pairs required. So you need a 30x coverage for this assay, and the number of libraries. So because, well, every library is exhausted after a certain amount of sequencing. So let's say if you're sequencing if you're putting together an organism with, let's say, 2.5 gigabases of genome size, then you are going to need probably 2 libraries. And if you have a more complex genome, then you're going to need more libraries. And these are the number of reads that you're going to need. So, okay, so I guess we're going to leave questions at the end. Thank you for listening. So I'm going to now hand over to Chris for to tell you about Bionano.
Thanks, Isabel.
Thank you. I'm going to make you host.
Host? Okay, cool.
Make host. Okay.
Cool.
You should be host now.
So let's go screen 2, and you can see my screen, and then I'll start sharing and make this proceed. How does that look? Can you guys see my presentation?
Perfect.
Okay, cool. All right, so I'm going to introduce to you guys Bionano, which is an optical genome mapping technology, which is sort of different from both Hi-C and single tube LFR, which would help more on the sequencing side of things. All right. So here's an overview of the chat. I guess we're going to talk about the challenges of de novo assembly, which I imagine most of you are fairly familiar with. Then we'll talk about optical genome mapping as a concept, and then about how Bionano system works, the data processing and analysis behind that, which is quite important. We'll look at a few published examples, and then we'll have questions at the end. But I've only got 15 minutes to cover this, and it's fairly technical, so I'll move real quickly. I apologize in advance if I miss anything, but hopefully I answer it as we go along. I'll just also quickly make myself a laser pointer. All right, so probably the biggest issue in creating genome assemblies is the levels of repetitive elements. And you can see with this example of plants that there are large numbers of repetitive elements. The wheat genome is sort of greater than 80% repetitive. Miscanthus is greater than 95%. Let me just look at this person. And then you can see that's made up of things like retrotransposons and DNA transposons. It doesn't really get any better when you move into animals. Humans, for example, only 1/3 of the genome is a unique sequence, whereas 2/3 are repetitive elements. So when you then go to sort of reassemble next-gen sequencing data, or making sense of these repeats, it's very difficult to differentiate between what's a genuine repeat and what is sort of a duplication of a library fragment. But beyond that as well, when you go to sequence these things, there are issues around sequencing and distinguishing between repeats in the actual sequencer itself. So being an optical technology, this doesn't actually do any sequencing. So you won't get the ability to see sort of SNPs and things like this, but it's really more a complementary technology to the sequencing. So the image you're looking at on the screen is from the Stlfr unit that Bionano has produced. And these long pieces, these long blue lines are actually DNA. And so there's a stain that stains the backbone of the DNA, and then there's a sort of a stain that stains particular motifs within the DNA. And you can see that each piece of DNA ends up with a pattern. And you can probably also see by the circles that there are large numbers of repeats in some of these where you can identify that. Some of these repeats, I think this one here is over 100 kilobase pairs in length. So you can probably tell that these pieces of DNA that you're looking at are actually fairly long. But you can actually see the DNA in this instance, given it's an optical technology. It's pretty rare these days to actually see the DNA you're working with. So it's pretty cool technology there. So this is the Bionano system, and it's made up of the unit that they call Stlfr, which actually does the sort of imaging of the molecules. It's also made up of computational servers and access servers, which run the unit and then let you do analysis, and some software called Access and Solve, which help you assemble things. There's also the Stlfr chips, which are an important part of that, and there's a number of different chip sizes to sort of suit. And then also there's a sample prep and labeling kits that are designed for specific types of sample types as well. So the workflow is this: you have to do extraction of long DNA molecules and then label the DNA so you can see those 7 base motifs. And then there's the linearization of your long DNA in the nanochannel array, which is sort of dependent on the architecture of the chip they use. And then there's the imaging sort of step as the molecules move through the nanochannels on your array. And then there's the genome assembly, and then a hybrid scaffolding step where you take in your next-gen sequencing data and map it back to your Bionano genome data. So we'll run through this real quickly just so you have a good idea. Probably the most important part of this is the extraction of long DNA, and sort of long is a loose term for most things. People talk about high molecular weight DNA, but I believe the cutoff for high molecular weight DNA is actually only about 50 kb as a definition. So for this one, we really want to have megabase length DNA. So if you've already got extracted DNA, it's probably not going to work for this. The process, I guess, of extracting DNA is quite rough on DNA, and as a result, you end up kind of fragmenting it. Even if you do something as simple as like pipetting, it can create a large amount of shear forces. So Bionano has created a method using agarose gel plugs, which kind of stabilizes the cells and DNA. So you put your sample in, you use an agarose plug to stabilize it. You digest the cellular content, cell wall and so on, and open up the internal part of the cell. Then you digest the agarose and you're left with the chromosomes themselves. And obviously that's an oversimplification, but that's kind of the process. So once you've done that, you then add a paramagnetic disk, and that binds your DNA, and you transfer that disk to a new tube and then elute the DNA from the disk. So you've left all the cellular debris and stuff behind, and you're left with clean, very large pieces of DNA. The next step in that process is labeling, and Bionano have actually created 2 different types of labeling, and their first one they made was called Nick Label Repair Stain, or NLRS, and it used nickase enzymes to insert the stain into the DNA backbone. The new one that they have created is called Direct Label and Stain, and that kind of tags instead of creating cuts or breaks in the sequence there. So the problem with the initial one that they created, obviously, is that if they had nicks on both sides of the DNA backbone, then you could end up with the piece of DNA fragmenting. And I'll show you an example of what this looks like. So here's a map that was created making nickase enzymes, and you can see that it's fairly fragmented. These are still very long pieces, so please don't make sort of that assumption that they're small because they're represented smaller. But by comparison, when you use a direct label and stain map, you're getting very long pieces of DNA. And in this instance that they're showing, it's probably the best example where you're getting chromosome arm length pieces of DNA. And this is the centromere where they've sort of broken in the middle. So the next step on the process is to run your very long pieces of DNA that have been labeled through the nanochannels. So this is the chip that you can use. This is a 3x, it has 3 wells, and you can also get 2x if you want. The chip itself has an architecture to it. It's a silicon chip, and there are sort of different sort of structures on the chip that help take sort of Gaussian coiled DNA into sort of relaxed and partially elongated DNA, and then fully into a single linearized piece of DNA in a single nanochannel. So this is an image of the Nano Array, and you can see all the single little lines. And this is an image from that array of single pieces of DNA moving down these channels and being imaged at the same time. Here's some pictures of the architecture that helps take coiled DNA and move it into sort of singular pieces of linearized DNA. There's 120,000 parallel nanochannels on that chip, and it's all made from silicon. And so people are pretty good now, thanks to computing, at making sort of accurate silicon chips. So here's a wee video, I hope it's going to come through, of DNA moving through the structures on the chip. And you can see it sort of gets works its way through the columns and into these relaxation channels, and then eventually going to see it move into sort of the nano channels, and you can see single pieces of DNA moving across the screen as they move through the nano channels. So as you're running your Sapphire unit, you can see real-time metrics on how it's going, so you know that whether or not it's sort of performing as you hoped, or if you've got good sizes of DNA, etc. And there's also elements of map rate, which I'll talk a little bit about later. So those single molecules of DNA are cycled through the nanochannel arrays and imaged. And there's just some things I need to clarify around this cycling that confused me when I first sort of started learning about Bionano. So I thought I'd do the same for you guys. This chip here has 3 wells, and each of these wells can cycle through 1,300 gigabase pairs of DNA. Each Stlfr unit can run 2 of these chips at once. And so we've got quite a large amount of DNA that you can sort of image if you keep running these. You need to generate a certain amount of coverage, which I guess is a carryover term from sequencing, to be confident in your, I guess, your map that you're going to build from these molecules that you're imaging. So these are some images that you, sorry, some data that shows what you're kind of going to really get, the levels of coverage you're going to need to get good mapping. So if we're looking at the hybrid scaffold side of things, with the Sapphire dual direct label and stain, you need 70 times raw coverage and a 60 times effective coverage. And so I'll break that down a little bit further. But the cycle that they're talking to really refers to the number of genomes that have been imaged based on the actual or estimated size of the genome. So for the example I can give here, the human genome is 3.2 gigabase pairs in size. So to achieve 100 times coverage of that size genome, you would need to image 320 gigabase pairs worth of DNA through this system, which would take about 8 hours. If you have a bigger genome and you wanted to get to that coverage, so if I keep going, you'd have to increase the length of time it ran, but I'll come to that further. The next thing you need to consider is how effectively the molecules of imaged map back to your genome. So there's a map rate measurement that the unit will keep track of and let you know. And you can figure out the effective coverage by multiplying the amount of filtered data by your map rate and then dividing it by the predicted genome size. So for your hybrid scaffolding or de novo assembly, the minimum recommended effective coverage is around 80 times, but we saw with direct labeling, you can get down to sort of 70 times. If we were doing the same example with human genomes, and you had an 80% map rate, then you'd be looking at this kind of calculation, would be 320 times 80% divided by 3.2, which gives you 80 times. So yeah, effective coverage versus actual coverage is something that's important to bear in mind. If you wanted to achieve higher coverage, you've got 2 options. You could run the unit for longer to cycle more of your genomes through the flow cell. Or if you wanted to sort of speed it up, you could run 2 samples in 2 separate wells and combine the data. And you just have to bear in mind that the effective coverage rate would still apply to that too. Yeah. So whenever you are imaging these molecules, the unit takes an image and converts it to a molecule. That's what they refer to it as. And then they line up their molecules based on the banding pattern of the 7-base motif stain. So you can see where all of these molecules have sort of been staggered across to build a map. They build this consensus genome map, and they represent it in the software as a sort of blue structure with lines across it that indicate where the where the 7-base stain was present. So if we keep going. So if you were looking for structural variants, then this would sort of tell you whether or not you've got insertions or deletions. And structural variants are more really of interest in sort of cancer studies, but I think now with conservation genomics, you're starting to see structural variants becoming considered in sort of breeding programs as well. But yes, that's useful. So, but for the guys who are here today, it's more about hybrid scaffolding. So you take your Bionano map and you would use your next-gen sequencing data, and this hybrid scaffolding pipeline that's part of the software goes through a process where it uses the same kind of labeling, but labels the 7 bases in your next-gen sequencing data, creates sort of an in silico map, and then lines it back up to the Bionano map that you've already got. And you can see exactly where these contexts line up to the map. So there's a particular software pipeline that you just have to sort of click that you want to run. And this is the process that it does automatically. So you generate those in silico maps by taking your next-gen sequencing data, scanning through the sequence for the 7-base pair motif, and then sort of creating a image that, sort of a digital image of that with a mark on it. And then you align those sort of in silico sequence maps against the genome map, and you start to resolve potential conflicts. And I'll talk a little bit about how conflicts are resolved. And then you merge those maps once you've identified and rectified the conflicts into a hybrid scaffold. And then you can kind of, once you've got your aligned maps, you can export them as an AGP or FASTA file for the scaffold. So in terms of your conflicts, it really depends on which, the system's going to look at which of those molecules has better coverage over the length of the conflict. If the Bionano genome map has a coverage over the entire conflict, it's going to go with the Bionano map. But if the sequencing data has better coverage at that point, it will go with the sequencing data. If you, the whole system identifies any points where there are conflicts and lets you look at them, links to them, and you can also manually resolve those if you want. So you've got a lot of control over how your resulting genome is fully aligned. So what this can do with Bionano data and combining it with next-gen sequencing data is can improve your contiguity by 1,000 times. That particular data came from some data that Bionano has offered, and they have improved it from about 0.08 to 0.09 megabase pairs up to about 50 to 80 megabase pairs for the EM50s of the assembly. So here are some species that have already been mapped. I'm not sure if they stand out to you based on what you're studying. I was quite interested in coffee, which was of interest to me. There's also been some use of this technology with the Vertebrate Genome Project. So they've started incorporating Bionano into their genome building, and probably for the Kiwis in the audience, you can see the kakapo, probably for the Australians, a snake. You've got lots of snakes. So what else can you do with this if you want to improve your contiguity further? The nickase enzymes that we mentioned about earlier can be used separately on 2 samples to provide a sort of a greater level of labeling, and combined using the next-gen sequencing data as kind of an intermediate step to improve the contiguity. You get a higher labeling density, and you can end up mapping shorter next-gen sequencing context back to your Bionano map as a result. So it doesn't really matter what sequencing data you put into this, you're going to get an improvement in contiguity, and some of these are quite large. If you decide not to do next-gen sequencing, you can still get very good maps, I guess, of the genome without that. This is the number of molecules with an N50 greater than 150 kilobase pairs. And this is the N50 of the map that's been generated by just Bionano on its own. But I think probably the whole ethos of this presentation is that combining these is going to end up with a better result. And here's some data that sort of shows that. So the initial NGS data that was provided for maize, kakapo, or blackbird, you can see that the N50 for that was 1.1 here, but when you combine it with Bionano, your assembly N50 increases to 100. And then with the kakapo from 4.34 to 71, and so on with the blackbird. When you look at the percentage of your next-gen sequencing data that's mapped to this, you can see that the percentages are very, very high. And here we have some different human reference genomes that were scaffolded using different sequencing technologies. So here we've got PacBio, Illumina, PacBio, and even old Sanger. And these are the Bionano scaffolding techniques. So here's one with the 2 enzyme or with multiple enzymes. And you can see that there's been an improvement in the the N50 of that genome based by incorporating Bionano into this workflow. So you can see it's gone from 17.92 and so on. If you were to look at this sort of from a workflow perspective, if you were to combine PacBio and Bionano, you could get a better assembly from 30 times coverage out of PacBio with 50 times Bionano than you could from just running 90 times PacBio on its own. And probably the biggest value there is that you don't have to do quite as much sequencing, which is very expensive on the PacBio platform. I know it's getting better though, so it's still a good value to talk about. So the largest genome that they've handled with Bionano has been 32 gigabase pairs. So, you know, it doesn't really bulk at big genomes. And what we're finding is that just in the last few months, we've had a large number of publications in high-end journals, and it seems that Bionano is becoming sort of required to get your data and your genome assembly into the high-end publications. So Nature Communications and Nature and so on. If I keep going, and we talk about the conclusions. Bionano genomic maps show the true structure of the genome. So you're actually looking at the physical genome you've isolated. What we found with the new direct label and stain labeling chemistry, that you can get up to chromosome arm length maps. Sorry, I've missed a word there. But if you combine your next-gen sequencing data, whether it's Hi-C data or Stlfr data and Bionano, you can actually get assemblies of the highest quality and further improve them if you decide to do Bionano with 2 enzymes on separate samples. Bionano improves your assembly no matter what sequencing strategy you originally used. And it's the only non-sequencing based scaffolding technology capable of correcting your sequencing errors that might come from that point. And including Bionano mapping data into your assemblies become a de facto standard and required for high level publishing. So that ran a little bit long. Thank you very much, guys. I'll hand it back now to Azadeh.
Thank you, Chris.
Participants. There you go. Thanks, Azadeh.
Thank you, Chris. Thanks, Chris. That was very interesting. So now I'm sharing my screen. So here, Chris and I would like to, we had a chat and we actually kind of put all these technologies next to each other and looked at how they can complement each other and what are the advantages and disadvantages of, or let's say shortcomings of every technology that can be complemented by the other one. So we've created this table based on our knowledge of the technologies, and we summarized different features of this technology in this table. We compared OmniC with Stlfr and Bionano in how effective they are in putting scaffolds together or contigs together, and what are the differences. So in terms of data range, so there are very big differences. That's, excuse me, probably the biggest difference between the technologies. So OmniC, as I mentioned, is both short range as well as
just as Dave's frozen on us there. So I guess what we were hoping to do was just a little bit of a comparison of the 3 options. Are you back, Azadeh?
Yes, I'm just getting water.
Okay, no problems. So you can see input material there for OmniC, fresh or frozen tissue, and for Bionano, fresh or frozen tissue. For Stlfr, we need high molecular weight DNA, and a bunch of different input options for OmniC and Bionano. In terms of contig orientation accuracy, Bionano obviously gives excellent accuracy. In terms of equipment needed to run the assay, so for Bionano, you do need access to the Sapphire instrument. We do have some options for that as well in terms of potential service around Australia if people are interested. In terms of cost, relative to each other, OmniC can be expensive. Stlfr is relatively inexpensive, and Bionano in the middle. In terms of SNPs, and phasing, Bionano is a no, but the other 2's a yes. Coping with repeat regions, yes, Bionano very good. With Stlfr, we just need ONT sometimes to gap fill. In terms of improvement in scaffold N50, I mean, Chris showed some good data on the N50 and Azadeh on OmniC as well. Azadeh?
Yes, I'm back. So yes, you can see that one of the major differences is the input. So if you want to kind of summarize the benefits of or advantages of every technology. Okay, so we have this other table. So the advantage of OmniC is that it produces even genome coverage. It produces both mid-range and long-range information in one assay. and it does provide an improvement over traditional high-C technologies in terms of coverage as well as accuracy of assembly. But the only matter is that you need fresh or frozen tissue. So if it's I don't know, an extinct kind of I don't know, if you need to have access to fresh tissue or frozen. Stlfr, you need very low DNA input. It can be already extracted DNA as long as it's long enough, just longer than 50 kilobases. It's good enough for Stlfr. There's no need for any equipment, and it provides a modest improvement. It provides an improvement in your genome assembly. You definitely see a much better contiguity in the genome, probably not so much like Omni-C or Bionano. And finally, Bionano, you can basically get chromosome length accurate maps. It's a non-NGS method, as Chris mentioned, and it can correct all errors that are introduced by common sequencing technologies. So we've seen cases where even PacBio assemblies, which are considered the highest quality assemblies, they actually sometimes you see these conflicts between Bionano map and PacBio map, and when the cases looked at more closely, it turns out that Bionano is actually correct, because what Bionano does is that you can actually look at the molecule. So it's a single molecule, which could be megabases in length, and you can see everything in one view. So a disadvantage of Bionano is that it doesn't provide sequencing data. So you can't get single base data information from Bionano. You have to combine it with NGS data to have both information, single nucleotide information, as well as correct contig and scaffolding. And you also need fresh tissue, so or frozen. So to summarize, what we would recommend is that if you're thinking about doing a genome assembly for your mid-range kind of part of the assembly, you can use either Omni-C or Stlfr depending on your budget. Stlfr is relatively budget friendly, and Omni-C produces for higher range data information, but well, it's a little bit more costly than Stlfr. We definitely, whether you use Omni-C or Stlfr for mid-range part of the assembly, we definitely recommend to include some Bionano mapping to create a hybrid assembly with the highest quality. So this is, I hope we managed to put all these technologies in a context that would be, make it easier for you to decide what's the correct technology or combination of technologies for your particular assembly. Do you have anything to add, Peter or Chris?
No, I think that's great. Any questions from the audience before we close the webinar? Got to leave an appropriate pause for unmuting. Well, thank you, everybody. We appreciate you joining us today. And yeah, as Azadeh said, we hope that was useful. We'll pass through the presentations, and we'll have this available as well as a recording. So if you have any questions, feel free to reach out to us. I know that you would have had Josh's email address when you registered. So thank you. Appreciate your time. Have a great day.
Bye.
Bye.